So how do scientists go about setting up these taxonomic figures, these phylogenetic trees? What's the information that they use to develop these? Well, that's what Concept 26.2 is all about. It says phylogenies are inferred from morphological and molecular data. And it says to infer phylogenies, systematists, man, I'm going to struggle over that one this whole chapter. Systematists gather information about morphologies, genes, and biochemistry of living organisms. So in any type of scientific endeavor, you want to try to gather as many streams of evidence as possible. If you get your evidence from a variety of sources in a variety of ways, you feel better about that evidence. You feel that that strengthens your idea or your hypothesis, and that's exactly what scientists do when they are trying to build these phylogenetic trees. They try to gather as much information and evidence as possible from a variety of sources. To talk about this, we need to kind of go back to a term that we've talked about in a previous chapter about homology or homologous structures when we were talking about evolution and maybe one or two chapters ago. Phenotypic and genetic similarities due to shared ancestry are called homologies, and we discussed that previously, like say for example the forearm bones of a bat, a whale, and a human all share some of the exact same number and types of bones and their placement. They do differ slightly in their size obviously and in the way that they're used, but they are homologous structures. Organisms with similar morphologies or DNA sequences are likely to be more closely related than organisms with different structures or sequences. So prior to the advent of DNA technology, the only thing we had to look at was the anatomy, the morphology of the creature. And this actually is a fairly good way of doing it, a fairly accurate way. But now we have not only the physical aspect of what we see about the creature, but we also have the molecular data of the DNA. And the same kind of rule applies. If there are a lot of similarities in the genetic data, then that usually means that the creatures are more closely related. The more genetic data that overlaps or that they have in common, generally speaking, the more closely related that they are. So the closer on a phylogenetic tree they would probably be. Before we had all this genetic testing, all that Linnaeus had to go by was the shape of the animal and the basic anatomy, perhaps through dissection and looking at its interior anatomy, things like that. And that turns out, like I said, to be usually a pretty good method, and that usually does match up even with the genetic data we have nowadays. Sometimes there have been discrepancies, and we go about correcting those whenever we get new data. One thing you have to be careful of when you're constructing these phylogenies, these trees, is that you have to be able to distinguish between analogy and homology, whether or not a characteristic or similarity is the result of common descent or whether it's analogous. In other words, is it because of convergent evolution? Homology is similarity due to shared ancestry, and analogy is similarity due to convergent evolution. And if you recall, convergent evolution occurs when similar environmental pressures, similar habitats, perhaps everything from similar weather conditions to similar land and environments, natural selection produce similar, what we call analogous adaptations in organisms that actually come from different evolutionary lineages. And the book gives you a good example of these two creatures, these two mole-like creatures. They both have an elongated body, they both have large digging front claws, they have small eyes, they have a pad of thick skin that protects kind of a tapered nose. They look very similar, and you would probably say that they are very closely related, although they actually are not that closely related because the top one is a marsupial, is an Australian, we'll call it a mole, and the bottom one is the Eutherian North American mole. The top one has a marsupial, it has a pouch, it gives birth to young that are not fully developed, quite a bit different than the Eutherian below, which is more like the mammals that we are familiar with here in North America that give live birth and produce milk and that type of thing. So these creatures have many analogous structures, although a marsupial and a mammal in this case, or a marsupial and a Eutherian, are not very closely related on the evolutionary phylogenetic tree of life. But they did come out to look very similar because they're under similar conditions. They both burrow in the soil, they both are well adapted to their particular habitats, which happen to be very much alike. So when we're looking at creatures, we have to be careful to distinguish the difference between homologous structures and analogous structures. A good example is a bat and a bird wing are homologous as forelimbs, but analogous as functional wings. You don't say a bat and a bird are fairly closely related because they both have wings, because if that was the case, you would put a butterfly as being more closely related to a bird and a bat than it is to, say, a beetle. So it's not the wings, because those are analogous structures. The forelimbs themselves, say the interior bone structure, that is what makes the bat and the bird more closely related. That is the homology that we're looking at here. Analogous structures or molecular sequences that evolved independently are also called homoplaces. Homoplaces is another term for these analogous structures. That word homoplaces comes from the Greek meaning to mold in the same way, so you can see why that would be a good word for analogous structures. Homology can be distinguished from analogy by comparing fossil evidence and the degree of complexity. The more complex two similar structures are, the more likely it is to be homologous. So if you look at the smallest of details and they still seem to be alike, then you can probably be more confident that they are homologous structures. Now, if we're going to use molecular data, genetic data, and we're going to look at molecular homologies or analogies, it gets quite a bit more difficult, quite a bit harder to do. Systematists actually use pretty advanced computer programs and mathematical tools to analyze comparable DNA segments from different organisms to see if they match up, to see if they have likenesses between them. So the first step to doing this is to sequence the molecules. After sequencing the DNA and getting it all laid out basically, then you have to try to align it, and it's like you're trying to line up this long chain of letters and trying to see if there are portions along the chain that sort of match. And so you can see where that would be quite tedious if you didn't have a computer to help you out to do that. If the species are very closely related, there's probably large chunks of the chain or many chunks along the chain where they are matching, or they differ only in one or two sites, one or two nucleotides. In contrast, if you're looking at two very different species, you're probably going to have a lot of different bases. You're not going to have as many likenesses along the chain. And this is because insertions and deletions accumulate over long periods of time, and as those species have diverged, the further they have diverged, the further their genetic code begins to look less and less alike. Now, one of the big difficulties in doing this is because you have deletions and insertions and you have various mutations that occur along the way, so you have to kind of use a computer program to try to take some of those into account. And in your textbook it shows you a good example of how these two particular creatures, these two species, they have diverged since their common ancestor, of course, and so there have been changes in their genetic code. But if you take into account some of the deletions and insertions, you can see where quite a few of their letters, basically, quite a few of the areas along their genetic code do match, but you have to use various types of computer models and mathematics to see this and to kind of work all this out. So I would think, even though this molecular data is amazing that it can give us, it sure seems like it's perhaps, especially before the use of computers, of course, it would be impossible. It's much more difficult than like looking at the creature and doing dissections and making your determination that way. Again, this chart, or this particular slide, is basically saying the same thing. It's without the mathematical tools and the computer tools that we have now, it would be pretty impossible to kind of sort through all of that genetic data to see whether or not these pieces of DNA, these strings of DNA, were homologous to each other. So this is called molecular systematics. Molecular systematics uses DNA and other molecular data to determine evolutionary relationships. And I think back again, I always do this when I'm talking about historical figures in science, and I wonder how much it would blow their mind to have the tools of today back in their time. I mean, I can't imagine what Linnaeus would think if we were able to describe to him and show him the tools that we have today to do classification in a way that he never would have dreamed of. Alright, let's take another quick break right there. So as you can see, there's a variety of basically streams of evidence that we use when we're developing these phylogenetic trees. And then one of the main things we have to be careful of is determining the differences between analogous and homologous structures, not only at the physical morphological level, but also at the molecular genetic level. We'll take a break. We'll come back with concept 26.3, which says shared characters are used to construct phylogenetic trees. So we'll get into the actual creation of some of these trees, what they look like, and again, some more terminology. Take a break, be right back.